Thanks. It's actually a real honour to be here, so thank you so much for having me. Like he mentioned, I'm going to talk a bit about some of the work that I've done. Actually, a lot of what I'm going to present I did in Belgium, so apologies for not showing anything too much new, but I'm happy to talk to any of you about some of the newer work that I'm doing as well. So I've called this title, this talk, Pubescent Plants, which sounds a bit odd perhaps, but I'm talking about how roots change their developmental patterns depending on the developmental stage of the plant. So the older the tree is that you take a cutting from, the harder it is to get roots to form. And what I'm going to show to you today is exactly when in that developmental pattern that decline occurs. So it's roots versus reproduction. And like Rob mentioned, I'm at the University of Nottingham in the Centre for Plant Integrative Biology at the moment. So I'm working on adventitious roots, and these are roots that form from any other tissue other than another root. So they're not lateral roots, but they're roots that form from the stem typically on cuttings. This is an example in Arabidopsis we can use as a model species. You can get adventitious roots to form along the hypocotyl, and I'll show you a bit more about that in a minute. In P, which is the system I work with a lot, it's a typical kind of cutting system, so the roots form from the base of the cutting. But adventitious roots are also really important in our crop species. So brace roots and crown roots are all adventitious roots. And I'm not going to talk about this now, but one of the interests that I have is looking at how these roots are different to these roots are different to these roots. Because everybody assumes they're the same, but actually I have some evidence in some of the work I've done that these are not the same. So are these going to be the same as well? That's a question that I'd really like to answer in the future. But enough about that. What I'm going to talk about today is peas. So like I mentioned before, adventitious roots are really important industrially for our crop production, in cutting propagation in the horticulture industry. All of you guys here will know very well about apples. So it's important for propagation of apples. And it's also important in forestry, which is my own background. And I'm going to talk to you a little bit about the forestry context. So in forestry, what they want to do is get a tree that has a wood property that's exactly what they want for their product. So what they do is they take seeds from a seed orchard. They grow the seedlings and they take cuttings from those seedlings and plant them out in hedges. And this is actually a commercial forestry nursery in Queensland. And what they do is they take cuttings from these, put them in the hedges, keep them quite low. And then you take cuttings from these hedges, so when you take the tops off, they grow new side branches, and that's what they use to take cuttings from. And then those cuttings are grown out and put into plantations. And then after some time, the trees that have the best wood properties for what they're after are selected out, and then the ones that they don't want are removed from the commercial hedges, so they don't take cuttings from those ones anymore. The trouble is, this process here to know what the best wood properties are takes at least 8 years. And by that stage, the cuttings from the hedges don't form roots anymore. So it takes a very long time, and it's not a very productive system like this. So you can see here, this is some work that I did in forestry before my PhD. When you take cuttings from 6-month-old hedges, so basically you take the top off, and then you take the side branches and make them cuttings. These form roots. You can see very nice root systems on these. But similarly, the commercial hedges, which were from the same parental seed lines, they don't form roots at all, and this is just 7 years. So you can see the significance of this problem. In forestry, what they do to overcome this is they'll take cuttings from their nursery seedlings, they put them in tissue culture, or they put them in fridge and cold storage. And this slows down the aging process, and then they can take cuttings from these later when they've decided which ones they want to keep. And they put these out then into the commercial hedges. But this takes a really long time and is very expensive for that entire time, and they throw out 90% of these. So it's a huge commercial outlay that they're doing for a problem that's quite severe for them. So I was working in forestry and looking at cuttings, and one of the things that I came across during this time is that we have a couple of problems working with woody cuttings. The first is that it's a very simple problem actually. We really didn't understand enough about how these roots form at all in the first place. So how can you work out what's going wrong if you don't even know what this process is in the first place? So that's the first problem. The second problem is if you're trying to improve this, if you're changing something like hormones, for example, which is my own interest, how do you know you're making an improvement if you've got no roots? You might actually be making an improvement, but you're just missing something else. So this makes it a really difficult system to understand in terms of improving root development. So this is where model species come in. During my PhD and some of my work now, I'm working with a couple of model species. The first is Arabidopsis, which everybody knows. It's a weed, I know, I agree. It looks like this when you put it in a pot, and it grows a floral bolt very quickly. If we want to form roots on them, what we do is, this is the seed here, we stick them in the dark and the hypocotyl gets nice and long. And then when you transfer them to the light, the adventitious roots form here. So you can see there's an adventitious root here, there's one there, there's one there. It's not very clear on that picture, but that's how we study the adventitious roots in Arabidopsis. In P, it's a much nicer system in terms of the cuttings, because you actually take a true cutting. So you grow the seed into a seedling, you take cuttings when you're ready, and then you look at the root development on the base of the cutting. So these two systems are great for different reasons. In Arabidopsis, we have lots of mutants. We can genetically dissect anything we want because we've got the genome mapped. The system itself is an easy screening system. So you put the seed on the plate, you germinate them, 2 weeks later you have results. So it's really fast, which is a little bit slower in P, but not a lot slower. It's a couple of weeks more. So you kind of double the time for the P system. The problem, to some degree, in Arabidopsis is that the adventitious roots are on an intact plant. They're not on cutting. You don't have a wound response, which you have in a normal cutting. But this can be a good thing if you're looking at the comparison, for example, to crops. So like I said before, the brace roots and crown roots on crops are all adventitious roots. And they're intact plants, so they're also not having a wound response. So maybe that's not such a bad thing, but not so good for this system. The trouble is, these roots are forming on the hypocotyl, and the hypocotyl has a tissue called the pericycle, which is exactly the same tissue that lateral roots form from. So some people argue that these are not true adventitious roots anyway, because they're forming from a similar tissue to what the actual lateral roots are forming from. Now, I've got plenty of results showing that they're not regulated the same as lateral roots, but it's a fair argument that maybe they're not the same as other adventitious roots. And the other problem with Arabidopsis is that the growth form is not really in a good system for taking cuttings or for looking at changes in developmental pattern, because this rosette forms a floral bolt very quickly. So trying to do something before flowering and after flowering in a similar system is really difficult in Arabidopsis. So in P, it's different. It has different advantages and disadvantages. So we have true adventitious roots. No one ever argues that. That's a cutting from its stem. It's very classical stem cutting propagation. The growth form is more appropriate, so it's more like a tree, which has branches and leaves and will flower at some point later in time. We do have some mutants available, and I'll show you some of those results in a minute. So that makes it quite useful for some genetic dissection. And it has a wound response, so it's more like a classical cutting. Again, that could be a bad thing comparing it to crops, but again, for this particular story, it was a very useful system. And like I said, genetic dissection is not as easy as in Arabidopsis. So I'm going to show you a lot of results about P, but many of the things that I'm showing you I've also done in Arabidopsis, but I'm going to just show you the results from P. So initially we had a question, and so coming from forestry, I was interested in cutting propagation. And we know that auxin, which is a plant hormone, is really important for root development and everything else. But in root development, if you have more auxin, usually you have more roots. And in P, we had a couple of mutants that had different auxin levels. So this is the level of auxin in the endogenous plant, so just that's present anyway. So this is the wild type level. And this particular mutant has 3 times more auxin produced in that plant. And we thought that would be a really nice model system to actually use to change hormone levels in terms of adventitious roots. And then we had another mutant here as a control, which has normal levels of auxin. So what I did was I grew these plants, I took cuttings, and I looked at the number of roots that formed on the base of the cutting. So this is the number of adventitious roots, and these are the same mutants as over here. And this mutant has wild type number of adventitious roots, despite having a much higher level of auxin. And this was a bit confusing for me, and I thought I'd messed up the line, so I did it a few times before I believed it. And actually what happens here is this mutant here with more adventitious roots has less strigolactones. And I'll tell you a bit more about strigolactones in a minute. And this one has a normal level of strigolactones. So actually then we started to ask the question of, well, is it strigolactones that's regulating adventitious root formation and not the auxin in this particular system? So strigolactones. Some of you may be a little bit aware of strigolactones because it's been important for mycorrhizae, but I'll go over the process here anyway. So it starts from a carotenoid precursor like ABA and several other pigments. And the carotenoid cleavage dioxygenases here, these two, they cleave the carotenoid precursor, and then through several other steps, they produce strigolactones. And this is the path that separates it from the ABA path. In P, the gene is called RMS5. So just to highlight that, because throughout the rest of this, I'll talk about RMS5. So it's a production part of the pathway. And then in order to have a response, there's an F-box protein, which is part of the receptor complex. There's other parts of that complex as well. And in P, that's called RMS4. And don't worry, I will put these labels on everything, so you won't have to remember that. And then what was known from the lab that I was doing my PhD in is that this whole process inhibits branching. So in order to get, in a normal tree, for example, you have an upright tree and then the branches only form in particular places. But if you block strigolactones, all of those axial buds will grow out into more branches. So you get a much more branched phenotype. Strigolactones, they were originally found for their signaling for striga and orobanche, which are parasitic weeds. So strigolactones are very unstable in the soil. So if a striga seed or an orobanche seed senses that there's strigolactones present, they know they're close to a root because it's exuded from roots, and if there is strigolactones, it has to be close to a root. So it's a signaling to actually germinate in these plants, which seems a bit odd because why would you want to encourage a parasitic weed from germinating close to your roots? But it turns out that this pathway is also very important for mycorrhizal branching. So if you have strigolactones, the mycorrhizae branch and will try and make an association with the root that they're close to. And just to show you the branching phenotype, this is a wild type P. These are actually leaves, they're not branches. But you can see here this is much, much bushier. And just up close, this is an axial bud that's growing out because there's no strigolactones present. So this is what was known at the time when I was looking at adventitious roots. So I grew my peas to about this big, so 2 to 3 leaves expanded. I took a cutting. And I took a cutting above the second scale leaf because previous literature looking at peas showed that this was a very good place to take cuttings. And then I'd put them in the base of, in a tube with either treatments or with water or whatever I was testing at the time to look at the root development on the base of these cuttings. These boxes we had made specifically so that the base of the cutting could be more or less in the dark. So it replicates a little bit more the soil environment. It's not a soil environment, but it's a little bit more similar to what we would do in a normal cutting system. And this is what an experiment would look like. So you can have all the boxes lined up, and then when you want to, you just pull the side off to have a look. And so what we found is that this is the wild type level of adventitious roots here. Again, this is the number of adventitious roots up the side. If you block out strigolactone production, so this is this one here, you block that out, you get more adventitious roots. And similarly, if you block out this part of the pathway here, the response part, you get more adventitious roots. So it looks like strigolactones are blocking adventitious roots because when you remove strigolactones, you get more. Just to prove this, we add strigolactones. So if you add strigolactones here and here and here, what you get is less adventitious roots in the wild type. You get wild type level of adventitious roots in the mutant that can't produce strigolactones but can still respond to them. And you get no change in the mutant that can't detect strigolactones, which is exactly what we expected. And just to show you what this looks like, this is a cutting of a wild type, and you can see there's an adventitious root here, there's some on the other side. And in RMS5, which is this one here with no strigolactone production, it's everywhere. The number of adventitious roots is much, much increased. And this is really typical. I could just pull out a cutting and know exactly what it was just from the number of roots it had. So it looks like this pathway is not only blocking branching, but also blocking adventitious root formation. So that's nice, but can we use it in a more applied way? Can we actually inhibit the production of strigolactones endogenously with a chemical to improve adventitious root formation? At the time, we didn't have any specific inhibitors, so we're blocking at this part, we're blocking all carotenoid production using fluoridone. And you see here that it's blocking pigment production. It will have changed ABA production, and to compensate for the ABA, we of course added ABA to show that it's not the same. And the plants have to be taken very careful care of because when they don't have the pigments, they can't respond to light, stress, and anything else. So if you take care of the plants, you actually do get an improvement in the number of adventitious roots, which was a really nice suggestion that this is going to work. So that's very cool. This again is the RMS5 mutant here. So it's not quite a full rescue, but we think that's because we're also getting it affecting other systems. Just to take that one step further out of a model system, we took a whole set of species, just cuttings from anything around campus at the time, This is just one example. In the paper, there's several other examples. And we added increasing doses of fluoridone. And this was done in just a dipping process. So just like you would do any other cutting from a forestry industry, we would just take the cutting, dip it in the treatment, and then put it straight in the soil. So this is soil experiments. And, oops, sorry. And this one is a commercial rooting hormone which has auxin in it. So you can see that at certain doses, you get to a point where this is the same positive effect as a commercially available rooting hormone. So this was really promising, and we're hoping that soon, maybe, they will get more specific inhibitors of strigolactones that only block this pathway and don't block other pathways, which would be really good. So that leads me to sort of first summary. So adventitious roots start by asymmetric cell division, just like in lateral roots, you get this process. But in between, we have a series of signaling pathways now. So you can see the rest of this in the paper in more detail. I've only talked about the strigolactone part, but there's other hormones that I'm going to show you briefly here. So you have So you have the production of strigolactones here to the response of strigolactones, and this blocks adventitious root formation. We think it happens by blocking auxin transport, but this is yet to be fully proven, which is why this is a dashed line. But we also know that auxin promotes adventitious root formation, and we've shown that this pathway here is completely independently of the strigolactone pathway. So cytokinin signaling inhibits adventitious roots as well, but completely independently of strigolactones. So that was kind of cool, but then we've now got an inhibitor of adventitious roots. Could this be the reason why we have a decline in adventitious roots with age? And this is what I took on to my last postdoc position. So the first problem we have when we want to look at age is we have to separate the different types of age, because what you've got a seed that germinates from an embryo to a seedling, to a sapling, which then will form flowers, and you get a whole lot of different problems, including height. So this is called topophysis, is positional effects. And height affects things like the amount of time it takes for water and nutrients and other signals to make it up the tree, but it also affects light. And a lot of the papers that look at maturation and adventitious root formation can't separate these very, very easily, from floral development to different physiological aging as well. So, and just to give you a very clear what I mean by some of these terms, because this is kind of important. In the literature, all these things get confused, and we've actually made a system that you can separate these things. So what we have as physiological age is this number of cell divisions since germination of the apical meristem. So this is sort of the number of cell divisions that have gone by when it lays down that next tissue. Chronological age is the time since that tissue was laid down. And ontogenetic age, which is phase change, is flowering, vegetative. So you start with embryonic, vegetative, flowering, senescent, for example. And then topophysis, I meant. And just to show you what that means is the higher up you get, the older physiologically the apical meristem was when, for example, this leaf was laid down. And likewise, the higher up, the less time has been since it was laid down. So chronologically, it's younger. So they're not completely inverse, because this one is also affected by stress. The number of cell divisions will change depending on stress environment. So they're not exactly an inverse relationship. And as you can see, the higher up you get, the more light you get, and other things affect this. So this is the system the definitions that I've been working with. And the reason I point this out is because a lot of the papers have confused these different definitions, and they can mean different things. So what I did was I developed a system with P where we can actually separate the physiological age and the chronological age and work out, and the ontogenetic age, and work out when exactly this change in root development occurs. So for example, you can take a cutting here, and in these different aged, different grown seedlings, and they'll have different sizes because you've got more nodes. They'll have the same physiological age because at the time when this was laid down, they were the same age, the same number of cell divisions. But they're different chronological ages, because this one was laid down more recently here because it's younger. This one's been much longer since the base of the cutting was laid down. We can also then decapitate them, so we take out the size effect, and we can also then take cuttings in different positions. So we actually separate these two processes. We can see the difference. And we can do this during vegetative phase, so before the apical meristem becomes floral, and also after and across that junction. And I'm not going to show you all the results because that's a huge number of experiments, but I'm going to show you before and after the transition to flowering. So what we've got here is, this is the system I'm going to show you, but we've done it in all the other ones as well. When you take a cutting at the base of each of these different size cuttings or plants. And if we look at days after germination, this is 10 days after germination, 14 days, so this would be 14, this would be 18. And the number of adventitious roots on the base of these cuttings. Now, up to this point, this is where the transition to flowering occurs. And in the paper, you can look at the experiment we did to show this. But this is the point where floral development starts. And you can see while they're during the vegetative phase, you get an increase in number of adventitious roots on the base of the cutting. And I've shown here the rooting percentage, because from a commercial perspective, that's kind of what they're more interested in generally. And they all root pretty well. You get roots on almost every cutting. But once you cross that divide, so here is the divide again at around 18 days, so before or after, and the conditions will change exactly when that transition occurs, but it's around here. You get a decline in adventitious roots at that floral junction. And again, the number of cuttings that will form adventitious roots decreases considerably as well. So maybe we thought at this point it's the switch to flowering that's specifically the response. And these don't have flowers, I'd like to say. You can't see a flower, but the meristem has switched to become floral. So what we did was we used some early flowering pea mutants, which I got from Jim Weller in Tasmania. And just to show you the flowering phenotype first, this is the percentage of the cuttings that either were just vegetative or had flowers present. So you can see here, this is the LE. Now, LE is a gibberellin deficient mutant, which all of these flowering mutants have been made in. So all the flowering genetic mutants are in an LE background. And you can see the flowering phenotype is like wild type, actually. There's no flowers on these at 14 days after germination. But these two early flowering mutants have flowers present. So you can see already the LF mutant has a lot of flowers, and the DNE mutant has some as well. And this is at the very beginning. This is at the early stage, the 14 days after germination. And if you go then to 22 days after germination, then the LE starts to get flowers, and the others are like lots of flowers are present. So we've got a nice system to test the flowering theory. So I'm just putting that here. That's exactly the same thing that's over here, so you can see the flowering phenotype. And now what we've got here is the number of adventitious roots on the base of these cuttings. So LE is the blue one, LF, so this one here, the middle one, is LF, and DNE is the other one. So you can see here the early flowering mutants produce less adventitious roots. And similarly, the percentage of cuttings that will form roots is also decreased. And then again, when you look at them later on in time, when you've got more flowers present, it's even further reduced. So this was a nice idea that the number of adventitious roots is linked to the switch to flowering. So that's the first thing. We've shown that now we have a system that we can actually separate the different stages of development on adventitious roots. And the next step then is looking at whether strigolactones is part of that pathway. So that was the question we were coming to from before. So this is what I showed you already. Strigolactones improve adventitious root formation, and I'm not going to dwell on that. I already explained that. So now, could strigolactones cause that age-related decline? So using the system that I've already explained, we did the same experiment. So this is 14 days after germination, and you can see the typical phenotype, where we get more adventitious roots on the strigolactone deficient mutants and the strigolactone unresponsive mutants. And as time goes on in wild type, we get a decrease in adventitious roots, which I already showed you. But we also get a decline in the mutants. So strigolactones are not necessarily the story, because if it was strigolactones, we would expect these to just be level. We wouldn't expect them to decline. RMS4 has a delayed flowering phenotype, and again, that's in the paper. You can have a look in more detail. And that's why we think this is not as reduced at this stage here. If we look at a bit later on, they're all gone as well. And again, the rooting percentages. And again, RMS4 has a delayed flowering phenotype. So that's all in the paper if you want to look at the flowering time experiments. So, okay, it's not strigolactones. This was a bit of a problem because that was what I'd proposed. So then I had to go back to the drawing board, and with some collaborators, we were thinking about what we should do next. So if it's not strigolactones, maybe it's some other hormones. So we did a screening of hormones together with my collaborators. So we grew cuttings 14 days or 30 days after germination. We took the rooting zone, so only the part of the stem that will form roots normally. We sent them off to my collaborators in Germany, and we measured a whole lot of different hormones, a whole suite of them. I'm only going to talk about jasmonic acid and auxin today, but in the paper is also the whole suite of cytokinins. There's like 6 different cytokinins we measured as well. So jasmonic acid is important for wound response. So when you wound something, you get a peak in jasmonic acid production, and you can see that here in the young seedling, you get this peak. This is the same as this bit here in the blue box. I've just blown it up so you can see, but this is the time scale here since cutting, and this is the amount of jasmonic acid present. So we took them at 30 minutes, well, at no minutes, 30 minutes, 6 hours, 24 hours, and 96 hours after cutting. And so this is the half an hour and the 6 hours, and you can see this initial peak in jasmonic acid production, which is what we expected to see. But in the base of the old cuttings, this peak is really reduced and it's delayed. So we thought maybe that was part of the story. So what we did was we added jasmonic acid to the base of the cuttings during the rooting period. So initially what I did was just 6-hour treatment. I've tried longer treatments as well, but the 6-hour one is the one that's most interesting because that's the initial where you would normally get a peak in jasmonic acid. And you can see here that if you add jasmonic acid, this is the concentration added, you do get more adventitious roots in the young ones. But you don't in the old ones. In fact, it looks almost inhibited. But that's tricky because the level's already really low. So I'm not going to swear my life on that being an inhibition, but it's very not induced. We don't get an improvement in root production in the older cuttings. So, okay, that's interesting. It responds to jasmonic acid, but it's not improving our root development. And again, this is the rooting percentage. Auxin was the other one that we were really interested in because auxin, as everybody knows, is important for everything, including root development. And again, in the young ones, we see a peak in auxin production at 24 hours, so a little bit later. And this is earlier than what people had previously been looking at. Sorry, at 6 hours, not 24 hours. And so this was really nice because we see a peak in auxin production, or presence, not production necessarily, but in the base of the cutting. And in the old ones, this is missing. And we thought, hey, this is cool. It doesn't quite fit with our results from the mutant that I showed you at the beginning that has high endogenous auxin, but maybe this is, in this system, maybe this is what it is. So I added auxin. Where is it? Here we go. And yeah, we can make the young ones produce more adventitious roots if we add auxin. Not really excitingly, not sort of dramatically. And the old ones, we can't make any significant difference, actually. So, can't improve root development by adding auxin. That's a bit odd. So I looked indirectly at metabolism of auxin. So I'm going to give you a quick rundown of the pea anatomy of the stem. So this is what a cross-section of a pea stem would look like. And just up close, this is a vascular bundle here. And in here is the cambial cell. So these cells will divide to form either the xylem or the phloem. And when you get adventitious root formation, what will happen is these cells here and on the edge here, they will divide and form undifferentiated cells, which will then form a callus, which then will produce vascular tissue within the callus, which will then form a primordia, which becomes a root. So this is the part that we were really interested in, is what happens here during the process of adventitious root formation. So xylem, phloem, and the cambium, they're the sort of 3 important things. And this is just to give you an idea of what it looks like, because I'm going to show you this again in a minute. So what I used was a DR5 GUS line that has been made in the Canadian group. And DR5 GUS is an indicator of auxin response. So if you have auxin present, this will come on, regardless of whether the rest of the plant will respond. This gene will show GUS expression. So this is, across here will become the young bases, so the bottom of the rooting zone of the young cuttings. And across here will be the old cuttings. And you can see this is a bit messier because the tissue is harder and it's harder to take clear cuttings, clear sections. And this is at the time of cutting. So initially we take a cutting and then I put these straight away into the liquid nitrogen, and then this is what it looks like. Well, not liquid nitrogen, sorry, straight into the GUS staining. And this is what we get. So it looks kind of the same. There's a bit of GUS expression here, and this is in the cambium, and that's kind of what we expect. 24 hours later, which is, if you remember, is about after the peak in auxin presence, we get a big induction in the GUS expression here, which is what we expect because we have more auxin present. And we don't really get any Gus different to the time zero, which is also not really not really surprising, because we didn't have an increase in oxen presence. And again, 96 hours later, we basically have Gus expression everywhere in the base of the cutting, and in the old bases, we start to see it as well. So, okay, that fits with our results for our endogenous levels of oxen, but what happens if we add oxen? So if we add oxen to the base of the cuttings, this is with 0. Again, this is the young bases, this is the old bases. This is with a lower dose of oxen. And you see here that and this again is at time 0. So this is at 24 hours, my mistake. This is 24 hours later after the base of the cutting has been taken. This was treated with oxen, this was not treated with oxen, and this was treated with 1 millimolar of oxen. Let me get that straight. So no oxen, 1 millimolar of oxen, and this is an even higher dose of oxen. Okay, we've done lower doses as well. And this is looking at the Gus expression 24 hours after taking the cutting. Okay, so I think I clarified my mistake there. All right, so in the young bases, you can see that it responds to the oxen to a point, and you get this increase in expression. And it's a bit hard to see in the colouring here, but this is actually Gus expression pretty much all in that region there. But in the old bases, the Gus expression, believe me, that's actually Gus expression somewhere in there. You have to take my word for it, because it's never very clear. But even if we add quite high doses of oxen, we don't see a response at all to the DR5. So the oxen is getting changed to something else. And it's only when we add the highest dose that we start to see this ring of oxen present in the cambial region. Okay, so these ones are not responding. He's kind of asleep. And these are the ones that have switched to reproduction. And these ones are the young ones that have not switched to reproduction. So just to sort of sum that up in a bit of a cartoon. So here we've got reproductive capacity means we don't get a response. Okay, so this is our pubescent plants. So we've got a teenager here, and this is oxen. Mum is oxen. And the teenager are the stem cells or the cambial cells, and mum's going, get up and do something, form roots. And he's going, I don't think so, I don't think so, I don't think so. So we can look at this at different times, and we can look at this in terms of added oxen. So if we add oxen, if we don't add oxen, he's not responding. If we add a low dose of oxen, he's not responding. If we add a slightly higher dose of oxen, he's still not responding. And if we add the highest dose of oxen, he's going, oh yeah, okay, I'll go to school now. So this is what's happening when we switch to the reproductive phase. So this is why I called my talk pubescent plants. Yeah, so what we think is happening is that the oxen is being converted, it's being metabolized, and then it's being moved away from the rooting zone as a non-active product, which is why we're not getting a response. And this is sort of the next step that I'm going to test in the other species. And just to sum up this, we've also done the cytokinins. If you want to look at them, again, go look at the paper for all the cytokinin responses. But what we have here is jasmonic acid. This is oxen. Oh, those are not very clear here. And then it's cycling through time. So half an hour, 6 hours, 24 hours, 96 hours. And this is what the response looks like. So this is the level of jasmonic acid in the juvenile bases. This is the level of jasmonic acid in the mature bases. This is the level of oxen. And this is representing the DR5 Gus response. And you see here the outcome is roots, and in this one the outcome is no roots. So we think that's pretty interesting. We're going to look a bit more at jasmonic acid and at oxen in terms of other species and what happens as they switch to flower development. So if I have sort of 3 take-home messages for everybody. The first one is that strigolactones are not responsible for the age-related decline in rooting, which is a shame because that would have been a really cool story, but such is science, right? Already that's an interesting thing to find because people weren't sure what was going on with strigolactones. We do have a delayed peak in jasmonic acid once they switch to flower development, and the oxen peak is delayed and reduced in older cuttings as well, and we think that's linked to metabolism of oxen. So I want to thank you all for your time, and I especially want to thank a few funding bodies. So at Ghent I was funded by Marie Curie. Here I'm at Nottingham, funded by Newton International Fellowship, which is a Royal Society Fellowship. I'm based in the Centre for Plant Integrative Biology. The strigolactone work was done at University of Queensland and was co-funded by the Queensland Government and Forests New South Wales. collaborators that I've shown the results for are in Germany, fantastic people. It was a really, really good collaboration. I'm currently working for Eve DeSpet and Malcolm Bennett, and Christine Beveridge was my PhD supervisor, and Jim Weller and Patricia Polowick kindly supplied different pea mutant seeds. And this is our team at CPEB. It's 4 different root groups all together, and so thank you all for your time, and I'll take any questions.